Good evening, welcome back to everyone. Tonight we're going to complete this lecture that we started last week on genes, chromosomes and DNA, and then we'll have one more lecture to complete for tonight. So we are in week 10 of semester, week 11 of semester. Okay, so we finished here last week. All right, so let's just revise a little bit more about what we know of DNA, that incredible molecule DNA that really determines everything about an individual organism. And it was very interesting in the 1950s. In the 1950s, there was a race on amongst scientists around the world trying to determine the structure of DNA. So they knew that DNA was composed of the 4 nucleotides that you're familiar with and various histone proteins, but they didn't understand the structure. And then finally, this model of DNA structure was proposed by 2 scientists, James Watson and Francis Crick in 1953. And this was actually based on analysis that was performed by another scientist, Rosalind Franklin. And what she did differently to everyone else was that she carried out this particular technique that you see here, X-ray crystallography. She carried it out using wet DNA rather than dry DNA, which is what everyone else was using. And she had this result that she hadn't really had time to analyse. And the story goes that that James Watson and Francis Crick, or rather one of their research assistants, actually got a hold of this image produced by Franklin's lab, and they were able to quickly decipher what it meant in terms of DNA structure. So quite a story behind that. If you're interested, you can certainly find lots of information online. All right, but what did they find? They found that DNA is a double-stranded molecule. And those 2 strands of nucleotides, of course, are bonded together, and then that molecule is twisted into a double helix formation, or into a helical formation. Each strand comprises a sugar phosphate backbone and attached bases, and each strand is connected to a complementary strand of nucleotides, and the bonding is non-covalent, that is, it is hydrogen bonding which exists between the paired bases. We know that the bases in DNA are adenine, thymine, cytosine and guanine, and we also know that each base, or rather each nucleotide, has 3 parts: a phosphate group, a sugar and a base. So that's all revision for you, and here's some more revision. I rather like this schematic of DNA. I think it's very, very clear, and we can see on this strand of DNA here, you can very clearly see that it's the sugars and phosphates that actually comprise what's called the backbone of that strand of DNA. And then the same thing happens here. Sugar phosphate groups are the backbone here. And then the nitrogenous base components of each nucleotide are bonded together by hydrogen bonds, which you can see by those dotted lines. This schematic also very nicely shows the distinct ends of each strand of DNA. So if we look at this strand here on the left, we can see that this is the 5' end, and you might recall that that refers to the free or 5' phosphate of this particular nucleotide here. So in other words, this particular phosphate can form a bond to something else. And then if we go to the other end of that strand, it's known as the 3' end of this particular strand of DNA, and that 3' end is referring to this hydroxyl group, this OH group found on this particular sugar. And we call that a free hydroxyl group because it's available to bond to other species. And then if we look at this complementary strand of DNA, we can see that the 3' end is up here and the 5' end is down here. So this is why they refer to the two strands as being anti-parallel. Okay. All right, a little bit more revision. Purines, the purines include A's and G's, pyrimidines are C's and T's. Purines have a pair of fused rings, whereas the pyrimidines only have one ring. A's and T's are connected by 2 hydrogen bonds, G's and C's are connected by 3 hydrogen bonds, and of course this is the basis of the base pairing rules that you're also familiar with. We've also seen that the bases are linked together within one of those strands of DNA by bonds called phosphodiester bonds. We've seen that nucleic acids, that is each strand of DNA, has 2 distinct ends. The 3' end has a free hydroxyl group, and that's found on the 3' carbon of the sugar. The 5' end of a strand of DNA has a free phosphate group. And this becomes important for the next part of this lecture. We also saw that the 2 strands of the helix are said to be anti-parallel. That is, the 5' end of one strand is directly opposed to the 3' end of the other strand. All right, so now moving on to the very important concept within this lecture, and that is the concept of DNA replication. So how does DNA build a copy of itself? But before we look at that, let's just put that into a little bit of context, and let's imagine a single cell. Single cell, there's the nucleus, and this could be an animal cell, and here we have a plant cell. And these individual cells could be the first cells that are formed, to begin the life of a new organism. So these cells are called zygotes, single cells. And in order for the organism to form, these cells have to divide to produce additional cells. So this cell will divide into 2, and so will the plant cell, and then they'll divide again into 4, and then again, and again. And now we have an embryo, an animal embryo, a plant embryo. But the most important process that each cell has to undergo before it divides into 2 new cells is to replicate its DNA. It needs to exactly replicate its DNA so that each of these new cells receive an exact copy of the DNA that was in that original cell. I'll just clear that. Okay, so how does DNA build a copy of itself? DNA is said to be replicated semi-conservatively, and that means that each of the 2 DNA strands will provide the template for building a new strand using those base pairing rules that you're familiar with. And this semi-conservative replication allows synthesis of new strands with high fidelity, very, very high accuracy. And the new DNA molecules that are formed consist of one old strand from the original DNA molecule and one newly synthesized strand. So let's look at semi-conservative replication here. Let's grab my pen. So you can imagine that this is occurring in that one cell. in that one cell. Let's say this is the original DNA, the original chromosome. We're just looking at one here, and this is the parental double-stranded DNA. And just note the base pairing rules that exist here. So A always pairs with T on the other strand, C always pairs with G on the other strand. Okay, so when it's time for DNA to replicate before this single cell is going to divide into two cells, those two strands of DNA will separate from each other. They separate. So here's one of the strands, the 5 to 3' strand. And this other strand here, the 3 to 5' strand, is this one up here. Okay, so they're the two old strands of DNA. DNA replication is actually synthesizing a complementary strand. So you can see that this new strand that's synthesized is complementary to the original strand there, using those base pairing rules. And the same thing applies to here. So here's our old strand of DNA, and here's the new strand of DNA that's been synthesized, the new complementary strand that's been synthesized. So what you should be able to see here is that this new double-stranded DNA, which consists of one new strand and one old strand, and the same applies to this one, they are now exact copies of the original or parental double-stranded DNA. So when this cell divides into two, each of those cells will have exactly the same DNA, exactly the same DNA as each other, and exactly the same as what was there in that original single cell. So that's the process that we'll be looking at tonight. Okay, and we'll be looking at specifically DNA replication in eukaryotes. So of course, animals, plants, fungi, and the protists. All right, so replication, DNA replication involves several very specific enzymes, Gyrases, these are enzymes that are required to unwind that supercoiled DNA double helix, and then an enzyme called a helicase is required to separate the two strands from each other. So the helicase enzyme can break those hydrogen bonds that holds the two strands together. All right, so let's have a look at that on the next slide. All right, so here we have, sorry, I need to just draw something. Okay, so you can imagine, I'll just draw it over here. So you can imagine that the DNA starts as this coiled, this double-stranded coiled helix. So that has to be firstly unwound at a specific region, in a specific region. So both strands are not just completely unwound at the one time. Part of the DNA is unwound, and then the bonds are actually broken between the two strands. So what we're looking at here is just one strand of DNA. One strand of DNA, and this one goes from 5' to 3'. All right, the first enzyme to come along, that is after the gyrase and the helicase, is this enzyme called a primase. And what the primase does is that it synthesizes a very short stretch of RNA, not DNA, but RNA. And this RNA is complementary to the DNA, nucleotides here. This short stretch of RNA is called an RNA primer. And what's important is the ends. So we can see here a 3' hydroxyl group and a 5' phosphate group here. And you can also see that these ends are opposite to the ends of the DNA. Okay, now the importance of synthesizing this little RNA primer is that the next enzyme that will come along and start synthesizing a complementary strand of DNA has an absolute requirement for this free hydroxyl group. Without the free hydroxyl group, then that enzyme, which is called DNA polymerase, cannot start synthesizing the complementary DNA strand. All right, but we have the 3' hydroxyl group in place. So now this enzyme called a DNA polymerase can actually come along and start synthesizing this complementary strand of DNA. So remember, this strand here is complementary to the original strand of DNA. And we can see the direction that it's actually moving in, synthesizing in a 5' to 3' direction. And this is called continuous replication, because the polymerase enzyme will just keep synthesizing without interruption in this direction here. All right, so what we have here is a little bit of a mixture, this new strand that's being synthesized. We have a bit of RNA here, but the rest of it's DNA. So of course this RNA needs to be removed, and that's exactly what's happened here, because we can't have RNA nucleotides in a strand of DNA. So that's been removed. All right, so let me go back now. All right, so new strand synthesis is performed by an enzyme called DNA polymerase. Polymerization requires a template to copy. That template is provided by one of the original strands of DNA. DNA polymerases require priming to initiate the synthesis of the new strand of DNA, and that priming comes about when that primase enzyme builds that little RNA primer, and that's usually about 5 to 10 nucleotides long. The RNA primer provides that 3' hydroxyl group. That's the important part here. DNA polymerase 1 will check each added nucleotide, or each added base, and also will remove those little RNA primers after replication. Then another polymerase enzyme called DNA polymerase 3 attaches the new bases only in the 5' to 3' direction, away from the RNA primer sequence. So I should add also, and I'll just go back to here. So when this enzyme called DNA polymerase is moving along the original strand of DNA, and it's actually building this complementary strand here, it's incorporating nucleotides using the base pairing rules. So that means that there are adenines and thymines and cytosines and guanines, a pool of these nucleotides that are available in order for the DNA polymerase to build this new strand of DNA, this complementary strand of DNA. Okay, any questions so far? No? All right, in that case, yes, you have a question? Ask away. What is the exon? What is the exon? Yes, we're not talking about exons yet. Did I mention exon? What is the exon? Where is the exon? That comes up in the next lecture. Oh, the exonuclease. All right, so 5' to 3' exonuclease. This is an enzyme that can actually degrade a molecule. In this case, it can degrade DNA or RNA. That's all that's talking about, exonuclease. And when it says exonuclease, it means that it's degrading from one end, as opposed to an endonuclease, which would degrade in the middle of a molecule rather than starting from one end. So you can see that the RNA primer sequence has been removed here, and that's the actual function of the exonuclease. And of course, a nuclease is an enzyme that degrades nucleic acids. Okay. All right, good pickup. All right, so we've seen that DNA replication of that one strand of DNA always proceeds, or sorry, any strand of DNA will always proceed from 5' to 3' on the strand being produced. So the one strand that we were looking at in the previous example is synthesized continuously, and we call that the leading strand. But the other strand of DNA is a little bit different. It's a little bit more involved, and we call the other strand the lagging strand. So to illustrate this, I'm just going to share my whiteboard. Okay, there we go. All right, so what we were looking at, so let's just draw this up like that. All right, so what I'm drawing here, double-stranded DNA. Okay, 3'. So we were looking at replication of the 3' to 5' strand, which is this one here. So what I've drawn here is the double-stranded DNA, but it's opened up here. This region has been opened up to allow the DNA replication to take place. And this strand is 5' to 3'. Okay, so each of these little lines represents the nucleotides on this strand of DNA. Okay, and I will just change that. Okay, and here we have the complementary strand that's being synthesized in a continuous fashion in this direction. Okay, so that will just keep going in that direction. All right, but this strand of DNA is a little bit different. So remember when we started, we had this free hydroxyl group that allows the polymerase enzyme to begin replicating the strand of DNA. Okay. All right, but let's see what happens down here. That wasn't a good choice of colour. All right, so these are the nucleotides on the other strand of DNA. And remember, the first thing that has to happen is that we need that little RNA primer to be synthesized. Let me see if I can pick a better colour. All right, so if we, let's put our RNA primer here. There we go. So that's 5' to 3'. So that means this will be the 5' end of the primer, and this will be the 3' end right there. All right, so that means that the hydroxyl group is over here. on this end, on this side of the primer. So when the RNA polymerase then comes along and uses that free hydroxyl group to start replicating the DNA, it'll start replicating. Oh, I've done this the wrong way around. I have done this the wrong way around. Yes, thank you for picking that up. So let's get rid of that. There you go. Okay, hydroxyl group goes in this direction. You were testing me. Yeah, I was testing you. Okay, so replication, sorry, was proceeding in this direction. So I've done it opposite to how the image was. That's why I've confused myself. All right, so if this was the hydroxyl group, then replication is proceeding continuously in this direction. Okay. All right, because that would be 5' there, and that would be 3' there. All right, so here it's in reverse. Here's the 3' hydroxyl group, so replication is proceeding in this direction. The polymerase will start adding nucleotides to synthesize that complementary strand using this free hydroxyl group. So the DNA itself will be replicated in this direction. Okay, there we go. All right, but the important point here, so it's going in the opposite direction, but what I haven't mentioned is that the unwinding of the DNA is taking place in this direction. Okay, so this strand is okay. It'll just keep replicating over here. But this strand of DNA is being replicated in the wrong direction. So what the polymerase does, it actually replicates just a small bit of DNA there, complementary piece of DNA, and then it'll jump back. Sorry, I should have drawn another primer. There's another little primer here. It will actually jump back, use this, another RNA primer to start synthesizing DNA here. So it'll actually synthesize these very short stretches of DNA, and you can see that there's multiple RNA primers that are being synthesized, and this will continue on as the DNA is unwound in this direction. Does that make sense, or do I need to start that again? I really mucked that up, didn't I? Does that make sense to everyone? Or should I start that bit again? No? No? Okay, what does everyone else think? Would anyone like me to try that again and this time do it properly? Just the last two sentences. All right, so back here to this one. So as I said, the double-stranded DNA is unwinding here in this direction. So this strand of DNA that's being replicated will just continue to be replicated in the same direction, and there's no disruption to that. But because the RNA primer here has the hydroxyl group on the other end, that means that the polymerase can only replicate in this direction, away from what's called the replication fork. So what happens on this particular strand is that we need not just one RNA primer to enable the DNA polymerase to replicate, but multiple RNA primers. So you can see one there, one there. As this DNA unwinds, another RNA primer will be synthesized, and so on and so on and so on. So when the DNA polymerase is replicating, this strand of DNA, it will replicate it only in short segments, basically in between the RNA primers. Does that make sense? Because it's always replicating in the opposite direction to this replication fork. Does that make more sense, Simon? All right, let me go back to the lecture. Okay, the multiple primers. Are the multiple primers 5 to 3 or to 3 to 5? Okay, so here's the primers here. Okay, and they go from 5' on this end of the primer to 3' here. Yep, because remember this strand of DNA that we're looking at here is 5' on one end and 3' on the other end. Okay, so that's opposite to the actual primer itself, the RNA primer. Yeah, and the others are the same. Yep, so that's the same. Yep. Okay. All right, I made that unnecessarily complicated. It's really quite straightforward. All right, so let's recap. So on that original strand of DNA, we saw that the complementary strand that was being synthesized is just synthesized continuously without any interruption in that synthesis. But the other strand, called the lagging strand, is a little bit different. So the DNA polymerase has to work discontinuously in the direction away from the replication fork. So remember, the replication fork is where that double-stranded DNA is actually unwinding and separating out to enable replication to take place. So the DNA on this particular strand is synthesized as a series of small segments. And to enable that to occur, the primase enzyme has to attach not just one RNA primer, but a series of RNA primers along that template strand as the double-stranded DNA unwinds. And the DNA polymerase then comes in and starts replicating the DNA, but it's replicating the DNA away from the replication fork. So those small segments of DNA that are actually being synthesized are called Okazaki fragments, after the Japanese scientist that first discovered this process. And those Okazaki fragments have to be joined up eventually. So eventually those little RNA primers have to be removed, and then they are filled in with DNA nucleotides, and then those short fragments of DNA are all joined together by another enzyme called DNA ligase. And as I said, eventually those RNA nucleotides of those little primers are replaced with DNA nucleotides. So that's essentially, or the basics, I should say, of DNA replication. And we can see the process there, this is from your prescribed textbook. It's quite complicated, and that's why I prefer to actually draw the process, just to simplify it. All right, so just very briefly, let's touch on the fact that eukaryotic cells contain a lot more DNA than prokaryotes. So bacteria, as I think I've mentioned before, mostly tend to have maybe one chromosome, which has to be replicated. But eukaryotic cells typically contain anywhere from 10 or 5, let's say 5 chromosomes, up to over 100 chromosomes in one particular plant species. So there's a lot more DNA that has to be replicated. And in order for this DNA to be replicated in a very timely fashion, there's not just one region where that replication actually initiates. There are multiple regions that start the replication process. And those regions are called origins of replication. And there are multiple origins of replication on each chromosome. At each origin of replication, there are actually 2 replication forks that form. I'll show you this in just a minute. And then synthesis of the new DNA proceeds from 5' to 3'. And yep, we can see that on this slide here. So this is a single chromosome, eukaryotic chromosome, double-stranded chromosome. We can see the end of the chromosome, a region of DNA at the end is called a telomere, and there's another telomere at this particular end. This region, roughly in the middle of the chromosome, is called a centromere. This is really important for the DNA in the cell division process itself. But all you need to take home from this particular slide is the 3 regions of DNA that have actually opened up to enable DNA replication to take place. Each of these 3 regions are called replicons. So we've got replicon 1, 2, and 3. And we can see the DNA is being replicated here and here. And this is occurring in each replicon. And then, of course, as we saw on the previous slide, there's not just one replication fork, but 2 replication forks in each of those regions of DNA. So normally when DNA replication is occurring in a prokaryotic cell with just one circular chromosome, there's normally just one origin of replication that forms. Okay, so here's a summary of the important DNA replication enzymes. So the primase enzyme synthesizes RNA primers, gyrase unwinds the supercoiled DNA helicase, separates the DNA strands from each other. DNA polymerase 3 adds nucleotides to the growing strands of DNA, complementary nucleotides. DNA polymerase 1 replaces the primer, the short primers, with DNA nucleotides, and DNA ligase joins the Okazaki fragments. And also note the names of enzymes normally end in ase, and you can see that in every case here. And that brings us to a discussion now about genes. So this is lecture 16, the genetic code. So let's start, everyone's familiar with genes, with the term genes. We know that genes basically hold the information, the code, if you like, that determines everything about an organism. But what's the nature of that code and how is it used, basically? So we're going to start firstly by defining this word gene. Can anyone give me an idea of what you think? How could we define very simply what a gene is? So obviously genes are found in the DNA, but what exactly is a gene? Who has some ideas about what a gene actually is? Coding segment of DNA, yes. What is it coding for? Coding segment of DNA. So that's very good, because we're saying that there are specific segments of DNA that are actually genes, and that is correct. That is correct. There are only specific segments of DNA that are actually genes. Pattern of attributes in a code. Yes. Passed on. Yes. Yeah, well, all DNA is passed on. Yes. DNA is passed on. But what about the gene itself? Think about a gene. What is a gene? How would you define a gene? It is a specific segment of DNA. What is it coding for? It is coding for something. Coding for traits. Yes, well, yeah, eventually, absolutely. I'm looking for something more at the molecular level. What do genes actually code for? Yes, they hold code, but what are they coding for? Proteins, yes. Many genes code for proteins. Yep, but do all genes code for proteins? So if we think about double-stranded DNA, so all these nucleotides, we've got all these nucleotides in these two strands of DNA, so just representing one chromosome here, but you might only have a little bit of DNA here that is actually a gene, and you might have that bit there that's a gene and that bit there that's a gene. You might have two genes very close together. So most of the DNA, in fact, is not coding for anything. Some of these, yes, may code for proteins, but some genes may code for various types of RNA molecules. They don't actually code for proteins, but they do code for RNA molecules. So a really good, simple definition of a gene is a specific region of DNA that encodes a functional product, some functional product, whether that product is a protein or various types of RNA molecules. All right, so that's a good start. Now let me just clear that and let's go back to what's referred to as, well, it was originally referred to as the one gene, one enzyme hypothesis. One gene, one enzyme hypothesis. Oh, that should be one. All right, so this hypothesis was taken from a dogma, which was referred to originally as the central dogma of gene expression, and at some stage it changed to the central dogma of molecular biology. And what that dogma proposed was that DNA, or rather the code found in DNA, is transcribed into a code in RNA, and then the code in RNA is translated into an enzyme. So this is what was originally thought, and that's because the early studies on gene expression were really investigating various enzymes in yeasts. So this is why this particular dogma was basically proposed. But the problem was with this, and this sort of came about with further study. Firstly, is that not all proteins are enzymes, and second, not all genes don't normally encode an entire protein. Genes may encode a polypeptide, and then two or more polypeptides may be required to come together to form a functional protein. Okay. And also we need to remember that not all polypeptides will go on to make proteins that are enzymes. There's different types of proteins. Of course, you've got structural proteins and transport proteins and so on. Okay, so the later dogma was changed, and we now know that DNA holds a code that is transcribed into RNA. That code in RNA is then translated into a polypeptide. So a single gene provides the genetic instructions for one polypeptide. But genes also encode different products that are not polypeptides. They also encode various types of RNA molecules, which we'll come back to. All right, so that means that there must be a specific relationship between the original DNA sequence of the gene and the amino acid sequence of this polypeptide. And when we're talking about the DNA sequence of the gene, we're talking about the actual sequence of nucleotides that are found in that gene. So for example, does the gene have this DNA sequence, A, T, G, G, C, C, A, G? Is that the sequence? So really we're talking about the order of the nucleotides in that gene sequence. Or does it have this sequence of nucleotides? Okay, G, A, and so on. Okay, so two different gene sequences there. All right, but regardless of that, there must be some sort of relationship between what we're seeing in the DNA and the final polypeptide that's synthesized as a result of that gene being expressed. So the process of converting that code found in the gene, in DNA, converting that information into a specific polypeptide that will go on to form a protein, that whole process is referred to as gene expression. And that code that's held within the DNA and the RNA, that particular code or that information is deciphered using what's called the genetic code. And we'll come back to that genetic code in this lecture. So let me clear all that. All right, but before we go on to look at that process, let's look at the transcribing of the DNA into RNA and then the translating of that code in the RNA into a polypeptide. It's a little bit different in prokaryotes such as bacteria compared to what happens in eukaryotes such as plants and animals. But we can have a little bit of a look at this. So we have a single cell here. Here's the nucleus. Here we have just a single chromosome, and unfortunately it looks like it's linear. It looks like it's a circular chromosome, but that would be a linear chromosome, and a little portion of that would be the actual gene that's being expressed. So that gene is used as a template to form a single-stranded molecule of RNA. Here it's called pre-mRNA or pre-messenger RNA. That pre-messenger RNA is modified to form a final functional messenger RNA molecule that's transported out of the nucleus into the cytosol of the cell. That RNA molecule then attaches, as you can see here, to a ribosome. And remember, we called ribosomes the protein factories of the cell. And that's because they actually decipher the code within that RNA molecule, and as a result, they synthesize a polypeptide, which you can see here in purple. All right, so this whole process is referred to as gene expression. Let's go back again. So when we go from the gene in the DNA to a complementary strand of RNA. This part of the gene expression process is called transcription. Then when we go from the code in the RNA to a polypeptide, this part of the gene expression process is called translation. Okay, transcription and translation, put them together and we have gene expression. Now, the same two processes occur in prokaryotes, but they occur very much at the same time. So the processes aren't divided between the nucleus, which is where transcription takes place, and the cytosol, which is where translation takes place. They both occur together in the cytosol, of course, because prokaryotes do not have a nucleus. All right, so let's do a little bit more revision. So RNA has the same basic structure as DNA, consists of a sugar phosphate backbone with bases attached to the sugar. But here are the major differences between DNA and RNA, which you're already familiar with. Let me just clear that. All right, so RNA nucleotides have a hydroxyl group on the 2' position of the sugar. They have a hydroxyl group. So this is the difference between deoxyribonucleic acids, in other words, DNA, and ribonucleic acid, in other words, RNA. RNA uses this base called uracil instead of thymine, and RNA exists always as a single-stranded molecule, so just one strand of nucleotides, compared to DNA that always exists as 2 strands of nucleotides. And this actually, in fact, makes RNA quite a transient molecule. It's very short-lived. In fact, as soon as it's synthesized, it starts being degraded. So it's a very transient molecule, but DNA is very robust, very, very long-lived molecule. So let's start with transcription. So let me just write this up again. So remember that central dogma of gene expression. DNA is transcribed into RNA, and then RNA is translated into a polypeptide. Okay, so we're just looking at this part of the process here, which is called transcription. So DNA will act as a template, or rather one strand of the double-stranded DNA will act as a template for the synthesis of RNA in a process called transcription. Only one strand of DNA is used as the template, and then just like we saw in DNA replication, transcription will proceed from 5' to 3' on the strand of the complementary strand that is being produced. In other words, if you have just one strand of DNA here, and this region here is the gene, represents the actual gene sequence, then we will have a complementary strand of RNA being produced. Okay, it will be complementary to the DNA. Okay, so that's the first part of this gene expression process. DNA, and here's the complementary strand of RNA. Okay. All right, so in this, this process of transcription, so forming this complementary strand of RNA, nucleotides will be added according to the complementary base pairing rules. But remember, in this case, we need to apply the base pairing rules between DNA and RNA rather than DNA to DNA. All right, let's clear that and look at transcription of DNA. Now, the enzyme that's used to catalyse transcription is called RNA polymerase. So don't confuse this enzyme with the DNA polymerase that's used to replicate DNA that we saw in the previous lecture. All right, so let's look at what we've got here. We've got 2 strands of DNA in blue, referred to here as duplex DNA. So just like in DNA replication, those 2 strands of DNA have to be separated from each other in order for an enzyme to actually be able to access the DNA and catalyse that process. And here is the enzyme, the RNA polymerase enzyme. All right, in this particular case, this strand at the bottom here, called the template DNA strand, is the one that's going to be used to synthesize a complementary strand of RNA, which is shown here in red. All right, so this part of the RNA has already detached from the template DNA strand, and let's just look at the base pair, the base pairing rules here. So where there's a T in the DNA, there'll be an A in the RNA. Where there's a C in the DNA, there'll be a G in the RNA. But where there's an A in the DNA, there will be a U, a uracil in the RNA. So remember, that's the difference in the base pairing rules here between DNA and RNA. All right, one more thing to notice, the DNA strand orientations. So the template DNA strand here, that's actually being used. Here we go. So we've got 5' on this end, 3' on this end. So of course the RNA strand will be the opposite to that, and it is 5' here and 3' here. All right, the direction of transcription is occurring in that direction. So once again, from 5' to 3'. So obviously this region of DNA that we're seeing here represents a specific gene. All right, RNA polymerases, unlike DNA polymerases, do not require priming. So that little RNA primer that we saw in the previous lecture, that's not required during transcription. All right, the polymerase, the RNA polymerase itself will bind to a specific sequence of DNA that exists just before the start of the gene. And that region is referred to as upstream of the gene. And that region of DNA is called the promoter region, and it contains a specific sequence. Every promoter region of DNA contains a specific sequence that the RNA polymerase recognises, and it binds to it, and then it starts moving along that strand of DNA, adding complementary RNA nucleotides. So the initiation of transcription occurs by base pairing of the first RNA nucleotide. Then elongation of that RNA strand will continue by addition of complementary RNA nucleotides until that RNA polymerase enzyme reaches a sequence of DNA, so after the end of the actual gene sequence, and that's called a terminator sequence. And once the RNA polymerase reaches that specific sequence, it basically disengages and it stops transcribing. So the transcribed region of DNA is called a transcription unit, and the RNA strand that's been transcribed is called a primary transcript. All right, so on this slide we can just see how that first nucleotide, that first RNA nucleotide is added. It's complementary to the DNA. So here's the DNA template strand right here, and the first nucleotide that's going to be transcribed is an adenine. So of course the RNA polymerase enzyme will bring in a complementary uracil nucleotide, and it catalyses basically the formation of the hydrogen bonds between those two nucleotides. All right, the next nucleotide in the DNA template strand is a cytosine. So the RNA polymerase enzyme will bring in a complementary guanine nucleotide. And then, of course, the enzyme will catalyse the formation of a phosphodiester bond between those two RNA nucleotides. All right, now, as we said earlier, genes encode functional products. And many of those functional products are proteins, but many of them are also different types of RNA molecules, functional RNA molecules. There are three major types of RNA molecules that are actually synthesized as a result of transcription of genes. Okay, the first one is called messenger RNA. This is always abbreviated to mRNA, and this is the type of RNA that's involved in this sequence. DNA is transcribed to messenger RNA, and then messenger RNA will be translated into polypeptides. Okay, so that's messenger RNA. So messenger RNA will actually hold the code that determines the amino acid sequence of the final polypeptide at the end of this process. But there are also many genes that produce what's called ribosomal RNA, otherwise known as rRNA. And ribosomal RNA is not this type of RNA that will go on to form a polypeptide. It's involved in the process, and we'll see how it's involved in just a moment. And then we have another major type of RNA called transfer RNA, or tRNA. All right, so let's just put that all into just one image so you can see or start to get some idea of how that works. All right, some double-stranded DNA. Okay. All right, and here we have a gene. This region here will be a protein coding gene. So it's going to be transcribed into messenger RNA, and that will be translated into a polypeptide. Okay, polypeptide. There's my polypeptide. All right, so here we have another gene, and this one's going to encode for a ribosomal RNA, ribosomal RNA molecule. And that ribosomal RNA molecule, or rather at least two of them, will actually form, together with various types of proteins, will actually form a ribosome. Will form a ribosome. Okay, and you probably remember from previous slides that it's the ribosome, the protein factory known as a ribosome, that is responsible for synthesizing this polypeptide. All right, I should have called this messenger RNA. All right, so protein coding gene, ribosomal RNA gene, and then here's our third major one. There are many different types of RNA molecules, but we're only dealing with three of them here. This particular gene encodes another type of RNA called transfer RNA. And this particular type of RNA molecule And it looks sort of like that. This particular type of RNA molecule will actually bring amino acids to the ribosome. So it's basically a transport type RNA molecule. It will transport specific amino acids to the ribosome in order to enable the synthesis of a polypeptide chain. Okay, so hopefully that sort of puts it all into some sort of perspective, and we'll be looking at those processes from here on. All right, very, very briefly, a few words about transcription in prokaryotes, such as bacteria. So as you saw earlier, in prokaryotes, transcription occurs in the cytosol, not in a nucleus, because of course these organisms lack nuclei. They only have a single type of RNA polymerase that transcribes all types of genes. And the transcripts that are produced, so the RNA molecules that are produced as a result of transcription, many of them are said to be polycistronic. That is, more than one protein is produced by a single messenger RNA molecule. That's very different to what happens in eukaryotes. Transcription and translation in prokaryotes often occur at the same time. So they are referred to as coincident or coupled. Okay, and you can see this occurring here. So transcription of a gene, or maybe more than one gene at the same time, it could be, say, 3 genes is occurring at the same time. Here is the RNA transcript that's being synthesized. And before transcription is even completed, ribosomes are already binding to the messenger RNA molecule, and they are already synthesizing polypeptides. Okay, so both transcription and translation is occurring together in prokaryotes. All right, in eukaryotes, it's very different. So transcription always occurs just in the nucleus of the cell. Eukaryotic genes are usually monocistronic, that is, each gene will code just for a single polypeptide. That is, if they are protein coding genes. The eukaryotic nucleus has an organelle called the nucleolus, which is the site for ribosomal RNA synthesis. So this is where you tend to find regions of the chromosome that contain ribosomal RNA genes. And this is where they are being expressed, where they're being transcribed to form ribosomal RNA. The 3 major classes of RNA, that is messenger RNA, ribosomal RNA, and transfer RNA, are transcribed by different RNA polymerases. So once again, very different to what happens in prokaryotes. All right, so transcription has taken place and a complementary strand of RNA has been synthesized. That is, it is complementary to that gene sequence that was in the DNA. But the RNA has to be modified. And these modifications, there are 3 major modifications that take place to the RNA, and these are referred to as RNA processing. So both ends of the RNA molecule are modified before that final RNA molecule is transported out of the nucleus and into the cytosol of the cell. All right, firstly, a cap is added. What's called a cap is added to the 5' end of the RNA. The name of that cap is a methylguanidine. It's just a modified nucleotide. And then to the other end, the 3' end of the RNA molecule, a poly-A tail is added. A poly-A tail, which is basically just a long string of adenine nucleotides. Okay, so that's They're the modifications to the two ends of the RNA molecule. But another important part of the RNA processing is referred to as RNA splicing. So what happens in many genes, particularly in eukaryotic genes, the DNA sequence of the gene is actually contains regions that don't hold any code for the final polypeptide product. So these bits of non-coding DNA have to actually be cut out. They have to be spliced out. And those regions of DNA that have to be cut out are called introns. They're called introns. So the regions of that DNA of the gene that remain are called exons. The regions that have to be spliced out or cut out are called introns. And so we'll see that on the next slide. No, not quite. Okay, we will look at that in just a minute. So in the cell nucleus, the DNA includes all the exons and introns of the gene. So that DNA is first transcribed into a complementary RNA copy called nuclear RNA, or pre-RNA, if you like. The introns are then removed by a process called RNA splicing. The splicing occurs at specific DNA sequences, and then the remaining bits of DNA, which are called exons, are joined together. At the end of the process, that we have our final mature messenger RNA molecule. All right, so let's look at RNA processing here. So here we have the gene. So we've got double-stranded DNA at the top here, and we have a gene. So here it is. Here is the gene. So these are the coloured boxes, represents a gene sequence. We've got our first three nucleotides shown here, ATG, and the last three nucleotides of the gene are shown here as TAA. All right, the coloured boxes represent introns and exons, okay? And when that gene is first transcribed, we can see that the RNA that is transcribed, that has been synthesised, is exactly the same at the moment as the DNA, apart from the fact that it's complementary to the actual DNA. Here we can see the promoter region. So this region before the start of the gene, this is where the RNA polymerase enzyme binds. Then once it's actually bound, it moves along the actual DNA and transcribes one strand of the DNA into RNA. When the polymerase enzyme reaches a specific region here downstream of the gene, then it basically dissociates. This is called the transcription termination site in the DNA. All right, so we have our primary transcript, or our nuclear RNA, or pre-messenger RNA. There's lots of names for it. And at the moment, it has the orange exons and it has the green introns. All right, so let's look at processing. Three major types of processing of that RNA. So first, a 5' cap is added. Methyl guanine is added to that end. To this end, we have a 3' poly A tail added. Okay, so there it is. All right, any number of adenines are added there. So that's RNA processing number 2. And then we look down here, RNA processing step number 3 is splicing. And now you can see these green introns, they loop out, as you can see here, and then they're finally cut out from that RNA. And that leaves behind just these orange exons. Those 3 exons, 1, 2, and 3, are joined together. And now we have the final mature messenger RNA. Where do the introns go? The introns are immediately broken down. The introns are basically broken down into individual nucleotides, which can be reused. All right, so it sounds really odd. Why have introns in the first place? That would take many additional lectures to explore what they now know about introns. And it seems, in fact, that without introns, and they still don't know a lot about their functions, but they are finding with more and more research that they are very important in terms of where the gene will be expressed, in what tissues, in what cells, at what time. So they do form really important regulatory functions. And there's also something known as alternative splicing, where depending on what the final what final product is required, that actually determines what part of the gene is actually spliced out. So it could be that this full intron is spliced at one time, and only a part of that intron or this intron is actually spliced out for a different application. So very, very complex. All right, so there's our final mature messenger RNA molecule. And you'll find here, once again, we've got 3 nucleotides that are shown here, AUG, and this just happens to be known as a start codon. So the start of the actual protein coding part of the RNA, but we'll come back to that. And then here at the end of the gene, we can also see 3 nucleotides, UAA, and this happens to be what's called a stop codon. So it's basically signalling that this is the end of the protein coding part of the gene. But we'll come back to that also. And you can see also that this protein coding part of the gene shown here is referred to as an open reading frame, and that's because the code within this region has to be read and translated into an amino acid sequence within a polypeptide. All right, so now we move on to translation. So, excuse me, does anyone need a break before we go on? I know this is pretty heavy. DNA transcribed into messenger RNA, and that is translated into polypeptide. All right, so we've looked at how DNA, or rather a gene within DNA, is transcribed into messenger RNA. Now we're going to look at this second part, which is the translation of the code within messenger RNA into the amino acid sequence of a polypeptide. All right, so messenger RNA transfers information into a polypeptide in the form of a code, and that code is defined by the sequence of nucleotide bases in the RNA. Each amino acid that's going to be incorporated into a new polypeptide is actually determined by each 3 nucleotides in the messenger RNA molecule. And each run of 3 nucleotides is referred to as a codon. So this is where the code lies. All right, now I'll show you that in a minute. So since RNA is constructed from 4 types of nucleotides, we know that. There are 64, that means that there are 64 possible codons. So remember, a codon is simply a run of 3 nucleotides. All right, where did that number come from? 4 types of nucleotides, so 4 by 4 by 4. Now, they know that 3 of these codons, 3 of these 64 codons, are called stop codons. And these actually specify termination of the polypeptide chain. All All right, that leaves 61 codons to specify only 20 different amino acids. So remember, in one of our much earlier lectures, we said that there were 20 standard amino acids that are used to build polypeptides. So why do we need 61 codons to specify only 20 different amino acids? Well, what you'll find is that most of the amino acids are represented by more than one codon. So the genetic code that we're going to look at is said to be degenerate. Let me just clear this. All right, and here it is, the genetic code. Excuse me. Here is the code. Everyone refers to the code in DNA and then the code in RNA, but here is what it's all about. Here is the code. All right, so let's say your messenger RNA molecule had a sequence C, had this sequence C, A, C, A, C. Let's say this was the first codon in your messenger RNA, CAC. Okay, here's the first nucleotide position of our codon, C. Second nucleotide position in our codon is an A. Okay, so we use these as coordinates. Go into there, and then we can find CAC. And here it is. So this codon CAC in the messenger RNA actually encodes this amino acid called histidine, HIS histidine. Let's say the next codon, the next codon is, what will I use? Let's say we've got an A, U, and U. So here's our second codon in the messenger RNA sequence, AUU. First nucleotide position, see there. Second nucleotide position here. So this brings us to this part of the genetic code, AUU, here it is. So this particular codon here in the messenger RNA will be translated into this particular amino acid in the new polypeptide that's formed, and this is isoleucine. All right, so that's basically how the genetic code works. And I'll just clear all that. All right, so it's also important to note, for example, if we look at this part of the genetic code here, we can see that there are 4 different codons, UCU, UCC, UCA, UCG. All 4 of those codons encode the same amino acid. This is serine. And you can see that they differ, the 4 codons only differ in nucleotide position number 3. Let's look at another example. Here's proline, is this amino acid proline, also encoded by 4 different codons. Then we get something like methionine, is only encoded by this one codon, AUG. Methionine, and this tends to be the start codon of most genes, the start codon. Therefore, the first amino acid in most polypeptides is methionine. And then you've got 3 different stop codons that you can see up here. 3 stop codons. So this will signal the end of the newly synthesized polypeptide chain. All right, so that's the basis of the code. Now we can look at translation, the process of translation. Is everyone good to go on? Or do you need a quick break? Happy to keep going? What about everyone else? All good? All right, let's keep going then. All right, translation. Let's look at the code, how that code goes from messenger RNA in the form of those codons, 3 nucleotide codons, into amino acids in polypeptides. All right, so during this process of translation, a ribosome will bind to the messenger RNA molecule. And the ribosome will move along the messenger RNA molecule and read its sequence, 1 codon at a time, starting from the 5' end, and it moves towards the 3' end. Each amino acid, as we've just seen, is specified by a specific codon, or maybe by 4 specific codons, depending on the particular amino acid. We're going to see that codons, that is the codon, each codon within the messenger RNA molecule is going to pair up with a sequence of 3 complementary nucleotides called an anticodon, and that anticodon is carried by a transfer RNA molecule. Let's look at that, hopefully. Very difficult to visualize. Here we go. All right, so let's look at transfer RNA molecules, which are encoded by transfer RNA genes. Transfer RNA molecules have an anticodon at one end. So here's a transfer RNA molecule. Always reminds me of a clover leaf. At this end here, there's a stretch of 3 nucleotides here, which are referred to as an anticodon. And that anticodon will actually determine a specific amino acid that attaches here to this hydroxyl group. So that means each transfer RNA molecule is specific for one particular type of amino acid, as I said, and that type of amino acid is determined by this anticodon here. So transfer RNA molecules are transfer molecules. They act as adapter molecules, that is, they attach to amino acids and they bring those amino acids to the ribosome, where that polypeptide synthesis is taking place. All right, so each transfer RNA molecule only binds the appropriate amino acid depending on which anticodon it carries, and then it's recharged after depositing its amino acid into the growing polypeptide chain. So this particular transfer RNA molecule has this anticodon sequence, UAC, at this end of the molecule. And the complementary sequence to that, which you would find in the messenger RNA, is AUG. Now, if you look up AUG on that table, the genetic code table, you would find that AUG is the codon for this amino acid, methionine. And sure enough, this particular transfer RNA molecule will only bind and carry this particular amino acid, this methionine amino acid. So right here, you can now see the link between that code carried in the messenger RNA, which of course originated from a complementary code within the DNA. So you can see the link now between the messenger RNA codon and the specific amino acid is going to be incorporated into the newly formed polypeptide. There's the link for you right there. All right, before we go on with the process of translation, let's just revisit our protein factories, our ribosomes. Each ribosome consists of 2 subunits, and these subunits are made up of various proteins, but also ribosomal RNA molecules. The 2 ribosome subunits are normally just referred to as the large ribosome subunit and the small ribosome subunit. And you can see them here. So here's the large one and here's the small one here. So remember, both of these are actually made up of several different types of proteins and at least 2 different types of ribosomal RNA molecules, which of course are encoded for by many ribosomal RNA genes that are found on the chromosomes. So ribosomes actually provide a precise method of aligning the codons found within the messenger RNA molecule and aligning the transfer RNA molecules that are actually transporting the amino acids to the ribosome. The ribosomes enable the alignment between the 2 to ensure that amino acids are synthesized in the correct order within the polypeptide. So we can see that when those 2 ribosomes bind together, and here we have one complete ribosome. Now you can see where the messenger RNA binding site is, along here. And when the ribosome is complete and bound to the messenger RNA, you can see that there are 2 pockets here that form. One is called the P site and one is called the A site. Okay, now we can actually look at the process of translation. How does translation occur? Okay, so remember, DNA, messenger RNA, transcribed into messenger RNA, and that is translated into a polypeptide. Okay, so we're looking at translation now. All right, the first stage of translation is called initiation. So the ribosomes will actually form from 2 ribosomal subunits. The transfer RNA, which carries methionine, will actually bind to the start codon in the messenger RNA. So I should say that the messenger RNA has now bound to the ribosomes, and the large subunit of the ribosome will bind to form those A and P sites. So let's look at that, because it never makes much sense until you actually see the image. So here we have initiation of translation. So let's just look at the left here. So we have in blue, we have the small ribosome subunit, and we have the messenger RNA molecule that is bound. Here we have the first codon, AUG, and we have the first transfer RNA molecule with its complementary anticodon. UAC is complementary to AUG. And because it's complementary, it means that this transfer RNA molecule can actually bind to the messenger RNA. Okay, what is it carrying? It's carrying the first amino acid, methionine. All right, now to the right here, our large ribosomal subunit has actually bound, and we can see these 2 sites, the P site and the A site, and our first transfer RNA is situated in the P site. All right, step 2 of translation. This is the elongation phase. And I'll come back to this in a minute, but first think we'll just have a look at these images. All right, so let's just look over here to the left. Otherwise, this can be quite confusing. So here's our first transfer RNA molecule, and it has bound to the first codon in the RNA. It's carrying the amino acid methionine. Now, our second codon that you can see in the messenger RNA is UU. That means that this particular transfer RNA molecule with the complementary anticodon of AAA can actually bind, and it has bound in the A site of the ribosome. It carries this amino acid phenylalanine. All right, now the next step is for a bond to be formed between those amino acids. And you might not remember, but the bonds that form between amino acids are called peptide bonds. What catalyzes the formation of this bond between the amino acids is really unusual, very unusual and very rare in the molecular world. That is an enzyme component of the ribosome, which is not unusual in itself, but the fact that this enzyme is actually one of the RNA molecule components of the ribosome. That is rare, to have an enzyme that is actually an RNA molecule, but that's in fact what it is. This RNA component of the ribosome is an enzyme. It's called peptidyltransferase. Some people say peptidyltransferase, and it catalyzes the formation of the peptide bond between these two amino acids, which essentially means that this methionine is removed from its transfer RNA onto the phenylalanine amino acid of this transfer RNA in the A site. So when that occurs, this transfer RNA is then can then leave. There it is here. It's empty, no longer has an amino acid. It can go off and be recharged. That is, it can pick up another methionine amino acid. All right, so that's the first part of elongation. All right, so let's just look at this transfer RNA here. It's in the A site, but when the ribosome moves to the right, so it's moving from 5' to 3' direction, that means this particular transfer RNA will now end up being positioned in the P site. And that's exactly what we see here in the middle. So here's the same transfer RNA molecule. There it is with its AAA anticodon. It is now in the P site, and it now holds two amino acids joined together by a peptide bond. All right, so because the ribosome has moved over, now we have this new codon positioned in the A site. It has the sequence CUG that enables this specific transfer RNA to bind because it has a complementary anticodon of GAC, and that is specific, or rather the CUG in the RNA codes this particular amino acid. So once again, you could go back to that table, the genetic code, and you could work this out. So this is leucine, the amino acid leucine. Once again, a peptide bond needs to be formed in between these, and that is catalyzed by the peptidyltransferase component, enzyme component of the ribosome. So in other words, these two amino acids will effectively be bonded to the leucine. The ribosome moves over again, so that now this transfer RNA now has 3 amino acids bonded together. This is the beginning of our new polypeptide chain, and the process continues. All right, now I'm just going back one slide. So now you'll be able to understand the elongation. step. So it begins with the formation of a peptide bond between the methionine amino acid and the second amino acid. The process involves the addition of a sequence of amino acids specified by the codons. As each new amino acid is brought into position by its respective transfer RNA molecule, then a peptide bond is formed with the preceding amino acid. Translation proceeds 5' to 3' along the messenger RNA molecule, and the polypeptide that's being synthesized is always synthesized from the amino end to the carboxyl end. So if this doesn't make sense, go back to the lecture where we looked at the general structure of an amino acid, and you'll see that there's always an amino side and there's the carboxyl side of the amino acid. So that's elongation. And the final part of translation is called termination. So the ribosome is moving along the messenger RNA molecule, and when it arrives at a particular codon called a stop codon, remember we saw 3 of them in the table of the genetic code, then the elongation process will stall. And it stalls because there is no transfer RNA molecule for stop codons. What this signals is that it's basically the end of the gene and therefore the end of that polypeptide chain. So proteins called termination factors will actually come into play. They'll remove the last amino acid from the transfer RNA that it's actually attached to. That is the chain of amino acids that are attached to that very last transfer RNA. The ribosome will then separate back into its 2 subunits and leaves the messenger RNA, detaches from the messenger RNA. So that's illustrated here. So this is termination. So the ribosome has moved along the messenger RNA until the stop codon or termination codon is positioned in the A pocket here. There is no transfer RNA molecule for this particular codon. So these release factors will then come into play. So here's the release factor protein, and it will enable the detachment of the polypeptide chain from this last transfer RNA. All right, so what happens? Transfer RNA molecule leaves. Here is our completed polypeptide product here. So this is the amino end, and this is the carboxyl end of that polypeptide. The large subunit and small subunits of the ribosome actually detach from the messenger RNA, and the messenger RNA molecule is rapidly degraded. So that means that genes have to be expressing various, well, thousands of messenger RNA molecules constantly because, as I said, they're rapidly degraded and you need this constant synthesis of messenger RNA to take place. So here are the 3 phases of polypeptide synthesis. Initiation is where we started, and then we moved on to elongation, and then finally termination. And that is the process of translation, and we have completed the entire gene expression process. All right, but polypeptide synthesis is only the first step in the production of mature protein. So you know that you might need 2 or more polypeptides to form a functional protein. And then the protein could be further modified by the addition of chemical residues. Proteins are then targeted. So they're not just produced and they don't just float around and and go wherever they choose. They're actually targeted to particular parts of the cell. They might be targeted to organelles. They may be proteins that have to be secreted from the cell. They may be proteins that are required to build or maintain cell membranes. And the way that they're targeted is that they contain a short sequence of amino acids, which are called signal sequences. And these signal sequences will actually bind specifically to specific receptors that are only found at the correct location within the cell. And that's basically all part of the targeting process. Proteins, any proteins that have to be secreted from the cell are sorted and packaged in the Golgi apparatus. All right, and that's all illustrated here. Basically, lots of examples here of where the final protein may be targeted. So this is one cell that we see here. Here's the nucleus within the cell. Here is one chromosome, let's say, and a gene has been expressed, has been transcribed to form RNA. Here we have an RNA, mature messenger RNA molecule, which has been processed. So that messenger RNA molecule is transported out of the nucleus into the cytosol. It's bound to a free ribosome. Here, translation takes place to form a polypeptide. Now, let's just say, for instance, that polypeptide is now a functional protein. Where may it be targeted to? All right, it might be a protein that's required within the cytosol itself, or it could be targeted to the this organelle, the peroxisome. It could be one of the enzymes that you might find within the peroxisome. It may be targeted to this organelle, the mitochondrion. So it might be a protein that's required in the outer membrane or in the inner membrane. It could be a protein that's found within the actual mitochondria itself. It could be targeted to the membranes of the chloroplast, or it may be required on the thylakoid membranes inside the chloroplast. The protein may be targeted back into the nucleus. So this could be, for example, a histone protein that's required as part of the packaging of the DNA. All right, possibly that original messenger RNA molecule may have been translated by this particular ribosome that's attached to the rough endoplasmic reticulum. In that case, then the polypeptide would be modified within the ER, transported out to the Golgi, and then it could be targeted to the lysosome. So this could be one of those, the protein might be one of the hydrolytic enzymes found within the lysosome, or the protein may be destined for secretion, okay, outside the cell. So lots of examples there of how and where proteins may be targeted to within the cell. So in summary, gene expression involves the flow of information from DNA to messenger RNA in a process called transcription. Messenger RNA specifies the amino acid sequence of a polypeptide. Polypeptide assembly occurs at the ribosomes. The process here is called translation. The genetic code is a triplet code. Each group of 3 nucleotides codes for an amino acid or signals termination of the amino acid chain. And proteins, of course, are targeted and transported within the cell by targeting mechanisms. And that completes our lectures for tonight. Any questions? Here is your very last assess tutorial. based on tonight's lectures, which of course we started last week. These tutorial questions are worth 5% and they are due next Monday. So that's your last, not your last set of tute questions, but your last set of assessed tute questions. Okay, so let's look at what is remaining in terms of assessments. Oh, here we go. Oh, here it is. I couldn't find it. Right, so this is the assess tutorial that's due next Monday. And here's your final prac report. So prac report number 2 is due on Friday the 29th. So this is the last day of semester that prac report. Acid-base titration is due on the Friday. So they're your remaining 2 assessments apart from the final exam. Okay, does anyone have any questions? And of course, everyone's probably seen by now the final exam schedule. So I'll just leave that up in case anyone has any questions for me.
Is the recording off, Doctor?
No, I will take it off now. All right.